this is an approach that will work. We replace all the damage, all forms of damage, even DNA damage, at once.
If you haven't heard of Jean Hebert yet, the following conversation might blow your mind and make you think completely differently about aging and what we can do about it. So Jean is actually way less optimistic about the trajectory of the longevity field than his peers. And it's exactly that, his pessimism, that has inspired him to develop a completely different approach and framework to solving aging. These days, John is working on curing brain aging, which I use this word intentionally, curing, and in a moment you'll understand why and how, and we'll keep it quite high level. So if you're interested in more of the details and his roadmap to achieving this, I recommend watching his recent presentation at the Foresight Institute, and I'll share it at the link below. And yeah, the conversation ends a bit abruptly. It will be followed by a second part, so stay tuned for that. And I hope you find John's work as fascinating and as encouraging as I do. You make the distinction between physical aging and biological aging. I've never heard that before. Can you clarify what they mean and what's the difference?
Yeah, for sure. I think it's an important difference because we ignore the physical chemical process of aging, thinking that everything is biological and we can fix everything, from a biological, metabolic, or enzymatic angle. So we know that there is physical chemical damage that accumulates with age in people over time. It's remarkably linear, and it occurs from the time we're made as a fetus or newborn. And these forms of damage occur in long-lived proteins, some of which don't turn over at all for our lives, some of which turn over slowly, and then once they accumulate damage, no longer turn over, so keep accumulating damage as well. And those are what we would call non-enzymatic forms of damage. So it's not something that was generated by a normal process in the body, and it's not one that can be reversed by an enzyme or a normal process in the body. And that form of damage is, sort of what we see when we see an old person. They're lost all their elasticity, and that does not just skin level. That's like all our organs, including our brain, that is degrading at that level. And no matter what we do to the cells to make them perform better, even transiently, and get measures of health that seem to be improved, we haven't impacted that slow, degradation of the long-term components of our body. And if we don't do that, we're still going to hit that precipice at about, 90, 100. If you're a real outlier, more 110 years old. But you will hit that precipice, and things will, precipitously go downhill at that point, because cells just cannot be happy in an environment where everything is falling apart. The structure, their signals. So that's what I mean by distinction between the physical chemical process of aging that is not biological, and that's something that is not neglected by all, but by most people looking to extend life in the longevity space.
So this physical chemical damage, it only happens outside of cells, correct?
It mostly happens outside of cells. There are some forms of damage to long-lived protein components that are intracellular. DNA is a good example. So in non-dividing cells, DNA is there for our entire life. It accumulates mutations that are, not regulated processes, not biologically regulated processes, and there's no way of reversing those either. And So that may also contribute to aging, although I think it's really mostly what's out of cells. Some would argue that DNA damage is a central driver of aging. I think over time it can be. It certainly is a driver of cancer with age, which is not good. But so yeah, there is some damage that accumulates inside of cells as well.
So, if I understand correctly, you're saying that most of the work today has to do with the cells, while you think that a bigger portion of the damage and what causes aging is actually the other kind of damage that happens outside of the cells that is underworked and underappreciated.
Yeah, exactly. I think most of the damage occurs outside of cells. It's something that is not easy to measure in the first place, and but thankfully some biochemists who are interested also in extending life a long time ago studied these forms of damage to our long-lived proteins and sort of catalog them. And there's no dispute that they occur. They abandoned the idea of beating aging because they thought there's no way we can address this kind of damage. I disagree. I think there is a way, and I think we'll get to that shortly. Um, but yeah, that's the damage that is neglected when the focus is on cell processes. You can improve a cell's processes as as much as you want, but it is intimately in communication with its environment, and that determines ultimately its function, its ability to survive, um, and lifespan.
It's interesting. I heard you say that there is a lot of evidence showing that if you put young cells in an older tissue, they will behave as old cells. And it also works vice versa,
Yeah,
which then it makes sense what you say that this is the central maybe problem, like the bigger problem. Um, and can you explain why it's impossible to fix that kind of damage to the lay audience, if you can?
I mean theoretically it might be fixable. It's just we haven't imagined how we might do that yet. Um, because of the complexity, you know, there we would need like hundreds of drugs that specifically target, these changes in proteins that we don't want with age, these physical chemical changes that occur without affecting, the normal covalent bonds or protein associations that we wouldn't call aggregates, but would call, heterodimers or multimers, and drugs that can fix breaks. That's actually hard to imagine how that could ever occur. And breaks occur in proteins like elastins and collagens. So these, so we, it's just hard to conceive how you could come up with these drugs that might be able to address some of this damage without causing more problems to the functioning of cells by you know, because drugs always have side effects, even the best ones we have, and so now we're talking about orders of magnitude more complexity and necessity for specific targeting.
Right.
Also, genes. A lot of people are using genes or epigenetic approaches like partial reprogramming, and that cannot work either because in our genomes we don't have encoded genes that can produce machinery that recognizes this damage, or much less can reverse it or deal with it. So then we're talking about inventing new genetic programs in our cells and delivering them to our cells. And so this is starting to get like in the realm of hard to imagine how that's going to work sooner than, say, a more straightforward approach of replacing tissues.
So just to understand, because we've all heard people, some Harvard professors talk about how we will be able to double our lifespan, live to 200. What is going on? Is it are we in denial? Are people in the longevity field actually worried about this aspect? Like how do people who champion these more mainstream ways of working on it, how do they view this problem, and how do you explain the passivity on that side?
Yeah, I mean, I know it's difficult for people who don't have much of a bio background to appreciate the all the the raw data from the literature and look it up and understand it and digest it. So it makes sense that you would depend in that case on the opinion of experts. So why do certain scientists who you would think would know better um, sort of either are not aware of this, these types of damage, this physical damage that occurs over time, um, or are ignoring it, knowing that it exists, um, to me is puzzling. I mean, I don't have a good answer. Um,
Okay, I really want to hear about the approach you suggest, but just beforehand, with the current approaches that are more common, what do you see as the best case scenario for working on aging? How far can we get?
with the current approaches,
Yes.
yeah, I don't think we get past the precipice when the all your extracellular material is degraded so much that you essentially have very little homeostasis left. You know, when you're elderly, a minor infection, or you fall, you break your hip. I mean, that could spell the end. And so we're all still going to hit that point if we're using non-replacement approaches to reverse the damage to not just the cells, but more importantly, to the whole tissue with the extracellular component being considered.
Would it help, though, compress morbidity and reduce suffering from age-related diseases? Is that
Yeah,
kind of the extent of what it can do?
maybe it can. So, that I guess I would refer to that as increasing average lifespan or compressing morbidity, exactly. And so there are, already, effective medicines for that increase health or likelihood of survival. But that's essentially what, all of the medical field has been doing up to now is compressing morbidity for all of us. So some of these approaches could add to that potentially, although none of them have shown great success in doing so yet. But there is that potential to do so.
And where did the idea of using replacement for aging come from?
It came from reading the literature. You know, I read a book a long time ago by Caleb Finch, uh, Longevity, Senescence, and the Genome, or something like that. It's a pretty old book, uh, but it had lots of good references, lots of good background. So I started looking up some of these references because I've always been interested in beating aging. Ever since I was a kid, and they were anecdotal. Oh, this accumulates with aging. Maybe if we target this, we can live longer. And then one thing after another, and no one ever seemed to get anywhere. And then this book led me to all these studies that show this non-biological damage that accumulates. I'm like, okay, this is a problem. Like, how can we possibly deal with this? And so you know, that's what led to the idea that we're going to have to replace not just cells but tissues.
So we need to replace tissues because, as we said, cells themselves they are influenced by their environment, and we can't just fix the cells.
Yeah.
Yeah. And you come from a background of genetics, which makes it especially interesting how you kind of changed your focus to replacements. So if you can explain a bit more about the process you went through realizing that this was a more effective way to go about things as your interest is in aging.
Yeah, exactly. So always been interested in beating aging, and at a young age, even in high school, was doing a lot of reading on molecular genetics and genetics, and thought, oh, DNA is the code of life. How could it not have the answer that we're looking for? And then also did a PhD in genetics at that point, but already realizing maybe the limitations of genetics. So my PhD was working with embryonic stem cells because of their tremendous potential um, to generate all the cell types of the body, and even make new bodies, new individuals from these cells. Um, so the transition was already happening at that point. And then I went on to focus on the part of the body that I would argue is the most important, the brain, and the part of the brain that defines us as individuals the most might be the neocortex, although it's certainly not the only part of the brain that does that. The other parts are important as well. So yeah, so that's sort of my journey to get to this point.
I heard you say that actually, in terms of the FDA, the kind of work that you want to do is much easier actually to get approved because it's nothing new in that sense, as opposed to some like more benign looking, aging drugs that need a much more
Yeah.
complicated path to approval by the FDA. So in the eyes of the FDA, you're actually more conservative in a way than other approaches.
Yeah, and I don't necessarily think the FDA is conservative. I think they try to be reasonable. They've made mistakes in the past for sure, and their motives have been in the a minority of cases misguided. But overall, they do try to do things the best way they can based on the science. And so for a lot of these drugs that people complain about, we don't not getting FDA approval, it's because I agree with the FDA. The evidence is just not very good to getting them approved for these, to for the purpose of reversing aging. Some of these drugs are very effective for certain conditions, health conditions, and they're used for that. They're approved for that. Um, but yeah, no, again, replacement being an old school approach that has been effective. Think of heart replacements, whether they're artificial hearts or biological hearts. Um, you have a heart that's failing, and you have a good heart, and you replace it. You're fixed. I mean, to some extent. And you were asking also the question of how you go about dealing with people that think this is a very radical approach. But you know, in part because it involves surgery. But surgery is like the oldest, most effective, medical treatment that we've been using. And to this day, it's probably is still one of the most effective approaches to medicine. In terms of removing things you don't want or adding missing things. It's not great with bacterial or viral infections, but otherwise it's an effective tool for lots of ailments, including for the brain. Brain surgeries occur every day in all medical centers around the world. So it's not really radical. And some individuals have multiple brain surgeries, for example, with tumors that recur or with intractable epilepsy. Um, these are almost standard approaches. They're not really radical. They're like old school, if anything. Um, but for progressive brain tissue replacement, which you would need to do in order to maintain self, now you're, upping the degree of difficulty for sure, and upping the risk of complications. And that's something we can't ignore. So this is not something that um, has been all worked out already. Um, so there's a lot of work ahead, but it is not something that is, sort of unimaginable, or that we don't have at least the technical basis for, that we need to improve upon and optimize for our purposes.
So, can you explain what it means to do replacement when it comes to the brain, and what are the challenges?
The challenges come from um, maintaining self identity and doing it in a way that doesn't disrupt function. And we have the evidence that this should be doable. Um, so we know that we can. So there's two parts to replacement. One is removing old tissue and then adding new tissue. So we have evidence in the clinic that you can remove old tissue without a loss of function, or continuity of function, or discontinuity of function. Um, and that comes from different types of lesion, but best studied ones are from slow-growing benign gliomas that, unlike glioblastomas, can be removed once they become a problem. And we've seen those occur, for example, in the language center, in the language center or personality center. And, sometimes they'll be monitored for a few years. Sometimes they go undetected for years until, a patient has a seizure or really bad headaches. They have an MRI. It's like, oh, you have this massive tumor that's eaten away your language center, or, your frontal lobes, your personality center. And this is in individuals, in their 50s, 60s, 70s. And there's absolutely no change to them, their personality or their language, that anyone has detected. And then the surgeon says, we should remove this, go in, take it out. Now they have a big hole where their personality, much of their personality used to be, or a big hole where their language used to be. And they're still the same person, or they can still speak in a way that is indistinguishable to how they used to be able to speak. And you know, despite having a hole where their language center used to be. And that's because those functions over time have moved to a different place. So the timing is one of the challenges here that we're faced with, because this level of plasticity is not instantaneous. So if you have a stroke here, or a stroke in your language center, those functions are gone. A stroke in your frontal lobes just completely changes your personality, and that's it. Or in the language center, same thing. You lose the ability to speak. Might be a little bit of compensation over time, regaining some words, but it's essentially permanent damage. So plasticity takes time and use. So the reason why the language wasn't lost in these patients, or their personality didn't change, is because those are things that were used every day by the patient. And so they simply, over time, get re-encoded into a different part of the neocortex. So that's the first part, is these clinical examples show that you can lose or remove tissue without a discontinuity of self, or personality or function. The other part of the equation is adding new tissue. And here there's a lot of challenge, even more challenge, I think. So adding new tissue, um, there's good evidence from, again, many labs showing that if you put young brain precursor cells in the old brain, they'll follow their normal developmental program, differentiate, and connect with appropriate parts of the brain that they would normally. If they're in the visual cortex, for example, they'll connect to visual brain targets. If they're in the actually motor cortex, if they're in the visual cortex, they'll connect to visual parts of the brain. So they extend and make connections that are appropriate for where you put them in. So that's a really good sign. Um, we and a few other labs have also shown that if you put them in the visual cortex, they also receive inputs from parts of the brain, in the sense that over time these new neurons will respond to light, which is also a good sign. So they can connect with the host brain and they can receive signals from the host brain that appear to be appropriate. But we're still far from being able to achieve tissue replacement because those initial good findings are not function. So just having neurons fire appropriately, connect appropriately, respond appropriately is not equivalent to encoding useful information and processing it in a useful way to the host. We know that for that to happen, you need the normal complexity of the brain tissue to be there. And just putting in a bunch of cells does not recapitulate that normal structure and internal connectivity within the graft. And so that's where there's a lot of work to be done, even though there is, some proof of principle that if we do it right, connections will be made appropriately with the host brain, and then you will be able to use that new tissue to encode information in a meaningful way. So that's something that we're working on in my group, and I hope more people will work on. Again, we're focusing on one part of the brain, the neocortex, but there are other parts that are important that I hope more people will start working on. So there's a lot of work ahead, but in principle, this is an approach that will work. We replace all the damage, all forms of damage, even DNA damage, at once. So we could really completely reverse aging with replacing old tissue with new tissue.
This is so interesting how when things happen progressively, then you don't lose the functions because they migrate to other areas. What happens in those areas? Don't they take over other functions that we need?
Yeah, I think that does happen. I think there's a limited capacity, for encoding information in our brains. It's a very competitive space. What doesn't get used gets lost. You can see that when you look at how elderly, healthy elderly that are for their age, cognitively sharp, they use greater and greater areas of neocortex for their routine daily functions that they think about every day, whereas younger people have much more compartmentalized functions. And so the older people, even if they appear to be, cognitively sharp, they are doing it at the expense of real estate in their ability to encode broader, cognitive functions and information. Um, so as you get old, you're literally running out of substrate to be functional. But it's amazing that plasticity. It's another great example of plasticity making the most of what substrate it has. It's just the nature of how the neocortex encodes information is very use dependent and very plastic that way.
And the challenges you described in the work in replacing brain tissue, what makes you more optimistic about those challenges than about challenges such as just fixing tissue, not by replacement?
Yeah, so that's a good question. So, the fixing tissue stuff, as we described at the outset, has the need for inventing a lot of new things, new technologies. The replacement approach is one that we don't really have to invent anything new. I mean, we didn't make the brains in our heads. It was made. We have the blueprint of how it was made during development. Right? We know if we do a high enough resolution analysis of the cell types and the extracellular environment of those cells by proteomics, we can get a very clear blueprint that already exists of what we need to achieve to recapitulate that developmental process in the adult host brain. So this does not sound like fantasy. This just sounds like hard work. We need to analyze this and
Right.
make it work. After that, it's surgery. And again, there are risks and complications that we can try to minimize, but it's not. Surgery is not something that we have to invent. It's not a new technology. Although, again, I think that's something that we want to minimize the risk of complications from surgery, and so improving that is also an area that I think is very important to pursue.
How would it work at scale? How many surgeons will we need to have trained to do surgeries to like all people getting older all day long?
Yeah, yeah. I mean, there's the scaling. There's the surgical approach, but there's also the source of the replacement tissue. If it's an engineered approach from iPS cells, where we make a fetal or an embryonic-like precursor tissue that we've shown works after grafting, that can be scaled as Uh, uh, again, not the easiest thing to scale, but there are already people working on this. There are already people scaling iPS cells, or simpler versions of replacement where it's just focused on the cells, or just focused on like extracellular matrices. Those are already being scaled. So putting them together, will require a little more engineering, but nothing new in terms of technological inventions. Um, so I think that if that's the source of tissue that we end up using, I think we can scale that. Um, there are other potential sources as well. So, some people are working on these synthetic embryos, so taking iPS cells, and instead of making the different brain precursor cell types, you just develop a synthetic embryo to a stage that's old enough that it has the brain tissue you want for grafting. And I assume that can be scaled as well. These are grown in bioreactors, so just more bioreactors, more of those, and of course reproducibility is a must, or at least if not a must, then you have to be able to identify the ones that grew the way you wanted, that you can use as a source tissue, but you should be able to scale that as well. In the past, we've used fetal cells to treat brain disease like Parkinson's. That's much harder to scale and much harder to control reproducibility, which is why most places stop using these fetal cells, even though they showed that in some cases they essentially cured the Parkinson's patients. But because of the lack of reproducibility in the source of cells, in some cases it made the patients worse. So they stopped using those. But
I was wondering about that. Yeah.
yeah,
And how do you imagine, let's just talk about the brain, it playing out? Let's say we're in a world where this technology exists. So around what age would a person start getting surgeries and gradually replacing their brain tissue, and how long would it take, and how many surgeries would it be?
I think ideally, there are factors that are going to impact the choice of age of onset for these, as well as how many surgeries you'll want to do. You also asked about the scaling of surgeries, which is something that I think is a good question as well. But we can, maybe I can quickly address that here. But you know, it would take a lot of training. But again, you could set up programs to do that. There's a lot of ways you can train surgeons in these approaches, and we could definitely push for that. Um, there's also the smart surgical robots are getting better and better, and so that's and I assume they'll just get a lot better in the next 10 years as well. So that's another avenue for scaling the surgical part. So the onset of a treatment would depend on how the rate at which you're currently losing, your cognitive functions, like how much every year. Is it worth it to you um, compared to where the risk of complications is at the time this technology is developed? If the risk of complications is really low, you might be fine starting, in your 50s, 60s, 70s. It might be a matter of choice. Um, if the risk of complications is still somewhat high, you might want to wait till you're in your 70s, 80s to start doing this. Um, so I think that's going to be the balance. And the number of surgeries also might depend to some extent on, what age you are when you start these. If you're starting later in your 80s, 90s, you might want a sort of an accelerated version of this treatment. Whereas if you're starting younger in your 50s, 60s, you might take the more conservative approach that would involve, maybe a dozen surgeries over the course of, 2-3 decades.
Mm-hmm. Okay. So you decided to focus on the brain because I get the impression that when it comes to the body and there are different ways to go about it, that seems like an easier problem. But can you kind of give an overview of what it would entail and what approaches right now you're thinking of as optional?
Yeah, so there's progress being made on I guess two broad different fronts. One is biological replacement and one is non-biological replacement. And they can both be broken down into like whole body replacement, either synthetic or biological, or more part by part replacement. So that's roughly the approaches that are being taken. For the synthetic replacement of whole body, for example, there's a group at Yale that's developing technology that's of interest. This is Nina Sestan's group, where they can keep brains at least metabolically alive for quite some time, not long enough to be of interest yet, but it's technology that's developing, but it's completely synthetic way of keeping brains alive. With that, of course, you'd have to incorporate more, brain machine interfaces so that you could actually interact with the world. Um, but those are also being developed, separately by other companies. There's
Right.
also biological replacement, which is more intuitive. We know new bodies live, 80, 90, maybe, 100 years. And so, we already have proof of concept for biological bodies. And those, can also be produced right now only with, you know, um, foster mothers that would be willing to produce these bodies, but also, they would have to be produced in a way that's ethical, so that there's no sentient being that's in these bodies. So these would be brainless or headless bodies that would be developed. This would be likely to be highly controversial, but it is technologically speaking, a way that could be used to replace the whole body. And then there's more, part by part approaches, where you can think of the synthetic heart as an example versus a donor heart or a heart grown in another species. Those are already being done today, so that's not that futuristic. Those technologies are implemented today, and of course for long-term use would probably need to be optimized. But broadly speaking, those are the approaches one can conceive of for the body.